All right, so here's the very end of chapter 17, amino acids. So the last couple of things we need to talk about are molecules that are derivatives of amino acids. And we've talked about these in a bunch of different ways before, but we kind of just didn't say that they were really amino acid derivatives. So we've talked about heme, and heme being really important in hemoglobin. And so we'll talk about its structure. We'll talk about nucleotides and some various signaling molecules. So heme is technically a prosthetic group. So it is not a functional thing by itself. It's there in hemoglobin. And we know that if we look at the structure of heme, that we have this iron atom in the very middle of the heme that's has coordinating bonds with the nitrogens and bunch of ring structures kind of around the iron. And the iron is what binds to the oxygen to stabilize it so that your red blood cells can carry it all around, right? So it is an example of something that has what's called a porphyrin ring. And you might have heard porphyrin before because there are a lot of disorders that are related to porphyrin. And this is actually, it's actually a little bit cool that if you have a mutation in some of these enzymes in porphyrin biosynthesis, you actually have some phenotypes that we think are the basis of what people call vampires, right? So sensitivity to light and receding gums so that you have really long, looks like you have really long teeth, things like that come from that. It's kind of a strange little thing. But heme is in hemoglobin, like we had said, but it's also in cytochromes, and because it's in hemoglobin, it's also in myoglobin. So kind of how do we get it? We have glycine and succinyl CoA that will condense together. You'll release a little bit of carbon, and you're going to make this precursor molecule, the alpha amino levinylate. And so a part of this happens in the mitochondria. And so at this point, we can ship this precursor molecule out and we go into the cytosol. And so this is one of those big enzymes that if you have a defect in this enzyme that allows you to start to make these rings that you can have some of the porphyrin disorders. And so we make one ring, and that's your like 1/4 of your porphyrin. And so you have porphyrobilinogen. So that's your kind of precursor molecule. And then you have to combine 3 of them together to get the 4th, and then to get 4 together. And then we ship that back into our mitochondria, where then we can add our iron to it. And now we have kind of a complete heme unit. And so you don't have to memorize this pathway at all. I just kind of want you to know that it's derived from amino acids, heme is, and what it's essential for. So that's good. So what I kind of want to talk to you about is how do we now break it down? We've made heme, right? But we've made like kind of a lot of heme. Think about your red blood cells and how many red blood cells you have, and how often we have to turn over these red blood cells and degrade the heme. And if you kind of think about it in terms of if you fall down and you get a bruise, at first that bruise is very, very purple, right? It's very dark. And so that's because heme itself is purple. And when heme starts to break down, it turns into biliverdin. Now, biliverdin is actually green. It doesn't have the iron in it. And so that's part of the color change. And so if you look at your bruise over time, it'll start to get lighter. But it kind of changes color. And at the very end of your bruise healing, which is your body kind of breaking down all that heme, it starts to turn yellow. And that's because the biliverdin turns into bilirubin. Now, normally your body will take that bilirubin continue to break it down in the intestines or in the kidneys, kind of either way. And it can either be excreted in the feces or it can be excreted in the kidneys. And so if you go kind of the kidney route, it turns into one molecule. If you go the intestine route, it turns into another, and they each have their own color. And this is kind of what gives feces and urine the color, right? But what's kind of interesting, which you've probably heard of before, is that sometimes babies are born and they have excess bilirubin inside of their bloodstream, and they're kind of like yellowish, right? Their skin is yellow, and so that's what we call jaundiced. And so you can actually just put them under a UV light, and that UV light will help the body really start to break down that excess bilirubin so that they don't look jaundiced. Kind of a little interesting side trip. So that's kind of like your breakdown of those molecules. But now we have to talk about some other amino acid derived molecules that are really, really essential for your body for signaling and things like that. The ketoccholamines, including dopamine, epinephrine, norepinephrine, those are essential for neurotransmission. So if you need to signal something, these are extremely important. So let's talk about that. They all come from tyrosine. So, and we know that we get tyrosine from phenylalanine, right? We don't make tyrosine ourselves, but we can get it by the breakdown of phenylalanine. So, by the processing of phenylalanine. So, if you take tyrosine, you can convert tyrosine, and I just want you to know this short name right here. This is L-DOPA. You do actually have to know this. So tyrosine gets converted to L-DOPA, and at that point, the L-DOPA can get converted into a couple of different things. And the one that's really, 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 really important here is dopamine. So dopamine is extremely important in your brain's processing of movement. And so if you have a mutation in the enzymes that process tyrosine and turn it into dopamine, you have a neurological disorder. You have Parkinson's. And so if you know of anybody who has Parkinson's, you can see that characteristic kind of shake that they have. And it's because they don't have enough dopamine. And dopamine is really there to kind of quiet that background movement that we all kind of have. And if you don't have it, you start to see that kind of shaking. And so what we can do as clinicians is we can give, you can't give them more tyrosine because they're missing the enzyme, and you can't give them dopamine directly because if you put it in the bloodstream, it won't cross the blood-brain barrier. But you can give them L-DOPA. L-DOPA will cross the blood-brain barrier. Blood-brain barrier. And then once it's internalized, as long as these enzymes are okay, the aromatic amino acid decarboxylase, then they can make their own dopamine. Now, it's not perfect, but it does relieve some of their symptoms for a certain period of time. And so they can convert that tyrosine, well, that L-DOPA, if you're talking about the patient, into dopamine, and then the dopamine can get converted into norepinephrine and epinephrine. And we know norepinephrine, excuse me, and epinephrine are important in our fight or flight response. So there's a lot going on that comes from tyrosine, just tyrosine itself. Another example of important molecules that are derived from tyrosine are, if you come down here all the way to the bottom, the melanins. And we know that there are different kinds of melanins, and each of the different melanins has their own pigment. So the eumelanins are black and brown, and the pheomelanins are red and yellow. And so everybody has kind of a unique combination of each of those. And so that gives us our hair pigment and skin pigment and all that kind of stuff. So Normally tyrosine gets converted and it goes through multiple pathways, and I don't care that you know all about that, but the one really important enzyme in this pathway is tyranase. And tyranase is essential enzyme to make all of the melanins. So if you have a mutation in the um, tyranase, excuse me, you have albinism. So that's exactly what this little guy over here has. He has albinism, and they can't make any kind of pigment. And so if you know of anybody who has albinism, they have to be extremely, extremely careful about going out in the sun because they have no pigmentation. They are way more susceptible to UV radiation. So they have to be very careful about putting on sunscreen, about wearing long sleeves as much as possible, hats, all that kind of stuff. They have to be extremely cautious about it. All right, so the last one we kind of want to talk about is cell signaling. And so nitric oxide is a very, very potent neurotransmitter. And we derive nitric oxide from arginine, from L-arginine. And remember, all of our amino acids are L's, sugars are D's, right? Okay, so you go through a two-step process with nitric oxide synthase. And you don't have to know all of the intermediates, but I do want you to know that we turn arginine into nitric acid. And so what does nitric oxide do? And it's really involved in vasodilation. And vasodilation is super, super important in controlling a lot of our body's responses. And so you have different versions of the enzymes that create nitric oxide. You have some in endothelial cells, you have some in neuronal cells, and you have some that are, we call them inducible. But regardless of where we find them, where we find the nitric oxide, it's got an extremely short half-life. And so what I want to do is I want us to look inside the cell and look at the pathway of where nitric oxide goes, and kind of what's the downstream signaling. And some of this, most of this, you really already know. We just never kind of said where the nitric oxide comes from. So we'll get neuronal signals, right? And so here's our nerve cell, right? We're propagating that electrical signal. And so right at the synapse, and it's really not necessarily a synapse, it's really a nerve cell connecting to a non-neuronal cell. And in this case, we're talking about the endothelial cells. And so right at that neuroendothelial cell junction, you're going to have release of these, and they'll come kind of down this way, of these neurotransmitters called acetylcholine. God, I could talk about acetylcholine for a couple hours, but acetylcholine gets recognized by the acetylcholine receptor, which you can look at this and you go, oh, that's a G protein coupled receptor, right? I kind of know how those work. We're going to exchange GDP for GTP, activate that G protein coupled receptor, which then goes on and activates phospholipase C. Phospholipase C, it's a lipase, it cuts something. That thing that it cuts is PIP2, creates those second messengers of diacylglycerol and IP3. IP3 goes on and mediates and activates release of calcium from the endoplasmic reticulum. So calcium comes out. Calcium binds to this complex. This complex here is calmodulin. That's that CAM, calmodulin. And then you have the nitric oxide synthase, and we have the little E in front of it because it is for the endothelial cell. So you activate this enzyme, the enzyme changes our amino acid of arginine into citrulline and nitric oxide. Nitric oxide will simply diffuse across the cell membrane. And so you have an endothelial cell on the outside, and then you have a smooth muscle cell that's right next to the endothelial cell. And you got to think about like blood vessels, right? So your blood vessels have this endothelial cell lining, and then it's the muscular lining, and it's a smooth muscle. Okay, so the nitric oxide gets picked up by the guanidylate cyclase. Wow, it's too late. Okay, so we activate that, which converts our GTP to cyclic GMP. Cyclic GMP, we know, is a second messenger. That cyclic GMP will go on and activate a kinase. The kinase is protein kinase G. Protein kinase G, if it's a kinase, is going to go and phosphorylate target proteins. And so the downstream effect of that is that you're going to, those proteins will be activated in order to relax and dilate your blood vessels. But nitric oxide, we said, has a half-life of like 5 seconds, right? So the signaling, as quickly as it comes, is over. And so in order to keep dilating those smooth muscle cells, I would have to continue to have neuronal inputs, because this cyclic GMP should very quickly also be converted back into GMP so that the whole signaling pathway shuts down. Now, we talked about this last semester, well, in Biochem 1, that we know about this drug, and the drug is Viagra. That Viagra prevents the cyclic GMP from returning back into the linear form into GMP. And so if we stop that conversion, our cyclic GMP will keep activating protein kinase G, which will keep phosphorylating, which will dilate your smooth muscle, which functions to enhance erections in men. But here's the deal. If you have somebody who comes in and you think they're having a heart attack, right? They're clutching their chest if it's a man, right? Men tend to have more of the typical symptoms, whereas women have very, very, very different symptoms for heart attack. But anyway, they're having a heart attack. So what do you want to do? The first thing that you can give them that's going to be extremely quick, you can give them nitric oxide in order to very quickly dilate their blood vessels. But if you are a man and you go in and you are having a heart attack, one of the first things they ask you is, are you taking Viagra? And the reason is, if you are taking Viagra, if you've taken it in the last like 4 hours, it is dilating your blood vessels, so your blood pressure is dropping. And we're going to give you more nitric oxide that is going to, as it comes in a nitroglycerin, that's kind of how it's sold as. But in the body processes it to nitric oxide. And so then we're going to give you more drug that's going to dilate your blood vessel even more. So your blood pressure is just going to tank. And if your blood pressure gets too low, you stop pumping blood to your entire body, and that's it. So it's very important, no matter who you are, to tell your doctor everything so that they can make sure that you don't die. All right. So that is the end of 17.